In 2019, a research consortium quietly published a paper that sent reverberations through the global genetics community. The study had set out to do something deceptively simple: take a set of ancient genetic markers most densely concentrated in the indigenous peoples of what is now Canada, and run those markers against population databases spanning every inhabited region on Earth. What they expected to find was a clean, predictable picture. The markers would be strongest in the Americas, would taper off as you moved toward Asia, and would effectively vanish by the time you reached Europe, the Middle East, and sub-Saharan Africa. What they actually found was something that none of the lead researchers could immediately explain. Populations from Lebanon tested higher than populations from Mexico. A community in the Welsh Highlands showed concentrations of these markers that rivaled those found in First Nations communities in British Columbia. An isolated group in Pakistan carried a haplogroup so closely associated with ancient Canadian Indigenous ancestry that one geneticist reportedly stopped the analysis and rechecked the sample labels, certain there had been a laboratory error. There had not been. The data was clean. The results simply made no sense. If you find this channel valuable and want to see more deep dives like this one, consider subscribing. Every view and every subscription tells the algorithm that this kind of content matters, and it is the only way this channel keeps going. Hit the bell so you never miss an upload, and let us get into what may be one of the most disorienting discoveries in the modern science of human ancestry. To understand why these findings shocked the scientific community, it is necessary to understand what geneticists mean when they talk about Canadian DNA. The phrase is a shorthand for a much more technical concept, and it does not mean the DNA of people who hold Canadian citizenship or who were born in the country as it exists today. Canada as a political entity is only about a century and a half old. Human beings have been living on that land for between 14,000 and 25,000 years, depending on which archaeological site you accept. When geneticists use the phrase Canadian ancestral markers, they are referring to the genetic signatures most densely and most anciently concentrated in the First Nations, Métis, and Inuit peoples whose ancestors arrived during the Great Migrations out of Siberia across the Bering Land Bridge in the terminal Pleistocene. The specific markers researchers were tracking included a cluster of mitochondrial haplogroups, particularly haplogroup X2A, and certain Y chromosome variants falling under the Q haplogroup tree, specifically QM3 and its downstream branches. These markers are not found at high frequencies anywhere else on Earth, and they are old, ancient old. Old enough that tracing them backward through time leads not to any recent population mixing, but to a founding ancestral group that geneticists call ancient Beringians. The people who sat at the top of the world, genetically isolated on a land bridge that no longer exists, before they finally pushed south and became the ancestors of every indigenous population in the Americas. The ancient Beringians are one of the most important and least discussed chapters in the story of humanity. During the peak of the last glacial maximum, approximately 21,000 years ago, a group of humans became effectively cut off from the rest of the human world. Glaciers blocked passage both east and west. This founding population sat in what is now Alaska and the Yukon, genetically drifting, accumulating mutations entirely their own, for perhaps as long as 15,000 years. Some models suggest the effective population during this bottleneck may have been as small as a few hundred individuals. When the ice finally retreated and the way south opened up, these ancient Beringians moved fast, reaching the southern tip of South America within perhaps 2,000 years of leaving their northern refugium. They were the first Americans, and every indigenous person in the Western Hemisphere carries their genetic legacy. But here is where the story fractures the expected narrative. A subset of the genetic markers that trace back to this ancient Beringian population also appear. at lower but statistically significant frequencies in populations that have no obvious route of contact with the Americas. And those frequencies do not follow any geographic gradient that makes intuitive sense. Haplogroup X is the key. Mitochondrial DNA passes from mothers to children without recombination, preserving an unbroken maternal lineage stretching back through time. Most of the founding haplogroups of the Americas, A, B, C, and D, are clearly Siberian in origin. They appear in East Asian and Siberian populations at high frequencies, and their presence in the Americas is easily explained by the Bering Land Bridge migration. Haplogroup X is different. In the Americas, the specific variant X2A is found almost exclusively in indigenous North American populations, with the highest concentrations in the Ojibwe, the Nucha-Nulth, and several other First Nations groups concentrated primarily in what is now Canada and the northern United States. X2A has never been found at meaningful frequencies in Siberia or East Asia, which means it could not have arrived in the Americas via the same route as the other 4 founding haplogroups. Where it came from is a question that has never been fully resolved. But haplogroup X, in its broader form as X2, appears in one other place with notable frequency: the Middle East. Specifically in the Druze communities of Lebanon, Syria, and Israel, in certain Algerian Berber populations, and at detectably elevated levels among Basque communities in northern Spain. The presence of an ancient North American genetic marker in a Lebanese mountain community is not a mistake. It is a real signal with a real evolutionary history, and explaining it has consumed significant portions of several prominent geneticists' careers. The leading hypothesis is that haplogroup X diverged from a common ancestor in Western Eurasia, or the Middle East, approximately 30,000 years ago. One branch, X2, remained in the Middle East.
Are those
North
differences
Africa, and parts of
actually
Europe.
meaningful
Another
at a
branch,
genetic
ancestor of
level?
X2A, traveled east, perhaps across Central Asia, perhaps through a northern route that bypassed Siberia's eastern lowlands, and eventually reached the Beringian refugium, where it became part of the founding population of the Americas. This means the X2A carried by an Ojibwe elder in Ontario today, and the X2 carried by a Druze grandmother in the hills of Lebanon, trace back to the same source, separated by a divergence event that happened before either population's continuous history began. The Lebanese Druze, who have practiced strict endogamy since approximately the 11th century, have preserved that ancient signal at unusually high fidelity. They are not descended from Indigenous Canadians, but they carry a piece of the same ancestral heritage, separated by 30,000 years of divergence. The results appear to make no sense. The biology makes perfect sense. Once you accept the timescale you are actually looking at, the Y chromosome side of the story operates through a different mechanism but produces equally surprising results. The Q haplogroup is the dominant paternal lineage among indigenous peoples of the Americas. In its specifically American branch, QM3, it is found almost nowhere outside the Western Hemisphere, with one persistent exception: certain South Asian and Central Asian populations, particularly in communities in Pakistan's northern provinces. Studies conducted between 2004 and 2022 consistently found QM3 and its downstream variants in Pashtun, Hazara, and certain Tibetan populations at frequencies impossible to dismiss as random noise. The explanation that has gained the most traction is that the ancestor of QM3 was carried eastward across Eurasia before the Beringian migration, leaving descendants in both the populations that continued toward the Americas and the populations that moved southward into Central and South Asia. A Pakistani community carrying QM3 today is not descended from indigenous North Americans. They are genetic cousins, sharing a paternal ancestor who lived somewhere in Central Asia before QM3 even existed as a distinct branch. before Canada was anything more than ice. When researchers began assembling the ranked dataset, ordering global populations by overall concentration of these ancient Canadian-associated markers, the results defied virtually every intuitive assumption about what the list should look like. The expected ranking would have placed First Nations Canadian populations at the top, followed by other Indigenous North Americans, followed by Indigenous South Americans, followed perhaps by Siberian and East Asian populations as secondary relatives. What the actual ranking revealed was a far messier, more interesting picture. First Nations communities in Canada did top the list, as expected. Certain Inuit populations from Nunavut and the Northwest Territories ranked extremely high as well. But the third highest concentration on the entire list did not belong to any Native American population. It belonged to a Druze community in the mountains of Lebanon. The fourth position went to an Ojibwe community in Ontario. The fifth went to a Berber group in the Kabyle region of Algeria. The sixth and seventh were First Nations communities from British Columbia. The eighth was a Basque community from the Pyrenees. It went on this way through the entire top 50, with ancient Canadian markers appearing and disappearing in a pattern that seemed to follow the logic of deep geological time, rather than any map of the modern world. The Welsh connection gained attention far beyond the academic genetics community because it intersected unexpectedly with a very old piece of folklore. For centuries, a legend circulated in Wales about a prince named Madoc, who supposedly sailed west across the Atlantic in approximately 1170 CE and reached North America, where his descendants merged with a local indigenous tribe. The legend was largely dismissed as political mythology by the mid-20th century. But when researchers processed haplogroup X frequency data from populations across the British Isles, certain communities in North Wales and the Scottish Highlands carried haplogroup X at frequencies substantially higher than the surrounding British population. The most straightforward genetic explanation has nothing to do with Welsh princes sailing to America. The elevated X frequencies almost certainly reflect a residual presence of pre-Bronze Age Mesolithic European ancestry that was never fully displaced by the subsequent waves of Anatolian farmers and Yamnaya steppe herders who reshaped most of Europe's genetic landscape. Those Welsh and Scottish communities had preserved, through geographic isolation and limited admixture, a genetic stratum that was ancient even when the Romans arrived. That stratum connected them, at enormous remove, to the same pre-dispersal population that eventually contributed to the Baronian founding lineage. The Madoc legend was mythology. The genetic connection it accidentally pointed toward was real. It simply predated Madoc by approximately 29,000 years. Polynesia enters this story through a separate and even more contentious set of findings. In 2020, a landmark study published in Nature examined the genomes of ancient and modern Polynesian populations and found unambiguous genetic evidence of contact between Polynesian peoples and indigenous South Americans, with the admixture event dated to approximately 800 years ago, well before European contact. The South American signal came primarily from populations ancestrally connected to groups in what is now Colombia and Ecuador. But the genetic trail did not stop there. Indigenous South American populations in turn carry the full suite of Beringian founding markers, both the X2A mitochondrial haplogroup and QM3. When researchers used the Polynesian admixture study as a lens and traced ancestry chains backward, they found that populations in Samoa, Tonga, and the Cook Islands carry a statistically detectable quantity of DNA that ultimately traces its deepest ancestry to the ancient Beringian population. Samoans are not closely related to Canadian First Nations peoples in any recent or direct sense, but they are connected to them through a chain of migrations and contacts that zigzagged across the Pacific over several centuries, depositing a faint but measurable trace of ancient Canadian ancestral markers in communities that exist in the middle of the ocean, thousands of miles from the nearest North American coastline. In the 2019 ranked dataset, several Polynesian populations appeared in positions that surprised every researcher who had not been closely following the Pacific admixture literature, ranking higher than European populations that lived on the same landmass as the ancestral source of the original migration. Geography, the study kept demonstrating, is a map of where people are. Genetics is a map of where they have been, and those two maps do not always agree. The Aboriginal Australian signal is perhaps the most disorienting finding in the entire dataset. In 2015, a study published in Science reported that several Amazonian populations, specifically the Surui and Caritiana peoples of Brazil, carried a genetic signal more closely related to Aboriginal Australians and Papuans than to any other population on Earth outside of other indigenous Amazonian groups. This finding was labeled Population Y, after the Tupian word for ancestor. It suggested the Americas had been settled not in a single wave from Siberia, but in at least two waves. One Beringian wave that became the dominant ancestry of most indigenous Americans, and an earlier wave that may have arrived via a coastal route and left its clearest surviving signal in certain Amazonian communities. The Population Y signal connects to Canada because every Beringian migration, regardless of which wave it belonged to, passed through the Canadian landmass on its southward journey. The ancient Canadian land was not merely a corridor, it was a filter. Populations passed through it, mixed within it, and diverged from it. And the genetic diversity of every indigenous population in the Western Hemisphere carries in some form the imprint of time spent in that vast northern territory. Japan presents its own complications. The Ainu people, the indigenous population of Hokkaido, and the northern Japanese archipelago are descended from the Jomon, the original inhabitants of the Japanese archipelago who arrived approximately 15,000 years ago, before the Yayoi migration from the Korean peninsula largely replaced them across most of Japan. The Jomon, and by extension the Ainu, carry genetic profiles that place them in close ancestral proximity to the populations that fed into the Beringian founding group. When researchers applied the comparative marker analysis to the Ainu, they found concentrations of Beringian adjacent markers that placed several Ainu communities higher in the ranked dataset than multiple indigenous populations from Central and South America. An Ainu community in Hokkaido tested for more ancient Beringian-associated markers than a Quechua community in Peru. This is not because the Ainu are more Canadian than Peruvian Quechua. It is because the Quechua population has experienced thousands of years of additional genetic drift and admixture since the initial Beringian dispersal, while the Ainu, isolated on an island archipelago, practicing endogamy for millennia, preserved the ancestral signal at higher fidelity. The ranking is a measure of genetic preservation, not geographic proximity. The Hazara people of Afghanistan and Pakistan occupy a peculiar position in this story. Descended in part from Mongol soldiers who accompanied Genghis Khan's campaigns westward in the 13th century, the Hazara carry a genetic profile that reflects both their Mongol ancestry and pre-Mongol Central Asian populations. The Mongol-derived component traces back to populations closely related to the ancestors of the Beringian migrants, the ancient North Eurasian and East Asian populations that were the immediate source of the founding lineage of all indigenous Americans. When the dataset ranked the Hazara, they appeared in the top 30 for Beringian marker concentration, above most Western European populations, above most Middle Eastern populations. A Hazara farmer in Bamyan province carries, by this particular metric, more ancestral genetic connection to the indigenous peoples of Canada than an average person from France or Italy. This is not a statement about culture, identity, or history. It is a statement about the age and provenance of certain segments of DNA, and about the fact that human beings moved across this planet in patterns far more complex than any simple migration map suggests. The controversy surrounding these findings has not been purely scientific. Some indigenous communities in Canada and across the Americas have raised serious concerns about how genetic ancestry research is conducted, interpreted, and communicated. These concerns are legitimate. Genetic science has historically been weaponized against indigenous peoples, used to question the validity of land claims, to challenge community membership determinations, and to impose Western scientific frameworks onto conversations that indigenous communities feel should be governed by their own traditions. Indigenous scholars have made the point, repeatedly and correctly, that genetic ancestry is one thread in the fabric of indigenous identity. A thread, not the whole cloth. A genetic marker does not make someone Ojibwe. Thousands of years of language, ceremony, land relationship, kinship, and continuous community membership make someone Ojibwe. The genetics illuminate the deep history of a population, but they do not define its present. And any researcher who treats them as a definition rather than a data point is making a category error with real-world consequences for real communities. With that crucial context in place, what the ranked dataset actually reveals is not a statement about who is and who is not Indigenous Canadian in any meaningful cultural sense. What it reveals is a map of ancient human movement, far more interconnected, far more circuitous, and far older than anyone previously imagined. The populations that rank highest for ancient Beringian markers are not all direct descendants of ancient Canadians. They are populations that have preserved an old piece of shared ancestral heritage because they were isolated enough or practiced sufficient endogamy to keep the signal legible despite the passage of tens of thousands of years. The populations that rank low do not rank low because they are genetically distant from the ancient human story. They rank low because their populations have been heavily admixed over the intervening millennia, and the Beringian markers, which were never dominant in those regions to begin with, have been diluted by the cumulative flood of other ancestries. The ranking is a measure of isolation and genetic drift, not of inherent connection or disconnection. The implications of this research for the broader science of human migration are significant. The standard model of the peopling of the Americas, a single founding population crossing the Bering Land Bridge between 15 and 20,000 years ago, has been under sustained pressure for at least a decade. The Population Y findings, coastal migration evidence from South American archaeological sites that predate the land bridge window, and the complex haplogroup X distribution all point toward a settlement history that was messier and more layered than a single clean migration. Some researchers argue for 2 founding waves, some argue for 3. A minority position holds that there may have been contact events between Pacific Coast populations and South American indigenous groups at multiple points over several thousand years, creating a network of intermittent connection rather than a single founding dispersal. What the 2019 ranked dataset added was a global-scale perspective. A view that said the story of the ancient Beringians and their descendants is not just an American story. Its fingerprints are on populations in Lebanon and Algeria, and Wales, and Nepal and Samoa, because the deep human past was not divided into the neat continental chapters that modern political geography suggests. What this research ultimately forces on anyone willing to sit with it is a fundamental reconsideration of what ancestry means at different scales of time. At the scale of a human lifetime, ancestry is family, parents, grandparents, the people whose stories you can tell. At the scale of a few hundred years, ancestry becomes history. At the scale of a few thousand years, ancestry becomes archaeology. Reinforces that at the scale of tens of thousands of years, ancestry becomes something else entirely. It becomes chemistry. It becomes the accumulated record of cellular division stretching back through time, past all human history and all human memory, to founding populations that were small enough to fit in a large village and diverse enough to carry within their genomes almost every major lineage that would eventually spread across the Earth. At that scale, the idea that any ranking of populations by Canadian DNA carries any implication for modern identity or modern politics collapses entirely. A Lebanese Druze community does not have a prior claim to Canadian territory because their mitochondria carry haplogroup X2. A Sherpa community in Nepal is not culturally connected to the Nucha Nulth because their genomes share an ancient Central Asian ancestor. The ancient signal is a scientific observation about the deep past. It says nothing about the present except this, that human beings have been moving across this planet for a very long time, in patterns no simple map can capture, and that the boundaries people draw between populations, between continents, between nations, are always newer than the DNA, always smaller than the story, and always far less permanent than the people drawing them tend to believe. The ranked dataset has been updated 3 times since its initial publication, as new genomic data from previously unsampled populations has been incorporated. Each update has reshuffled portions of the ranking and added new surprises. In the 3rd update, published in early 2022, a previously unsampled community from the highlands of Eritrea appeared in the top 100 for haplogroup X frequency, a finding that generated a spirited debate across the published correspondence sections of 2 major journals. In the same update, in Sastoy, several communities from the southwestern United States dropped in the ranking as new full genome data revealed higher levels of recent admixture than earlier haplogroup-only studies had suggested. The ranking is not fixed. It is a snapshot of scientific knowledge at a given moment, constantly revised as the data improves and the models grow more sophisticated. What remains constant is the underlying reality it is trying to map. The ancient, complex, deeply interconnected story of where human beings came from and how they spread across the face of the Earth. The scientists who first saw these results and reached for their sample labels, certain something had gone wrong, were not making a mistake born of ignorance. They were making the very human error of assuming that the world is smaller and more legible than it actually is. The genetics kept correcting them, as it always does, with the patient authority of 60,000 years of accumulated mutation. The Lebanese grandmother and the Nunavut elder share a piece of chemistry. The Hazara farmer and the Ojibwe trapper are, at a depth of time no culture reaches, relatives. None of them know it. None of them need to know it for their lives and identities and communities to be real and whole and valid. But the knowledge is there, written in the molecules, waiting for the tools that could finally read it. The results make no sense by the map of the modern world, by the map of the ancient world, the one that existed before national borders, before continents had names, before human beings had language complex enough to ask where they came from. The results make perfect, extraordinary, humbling sense. The planet is older than our categories. The DNA has always known that. We are only now learning to listen.